So yesterday we talked about like the hardwired connectome and how it can achieve plasticity and all that. So I stopped here there with this slide, and let's begin with this slide. So we talked about how the network develops over development, how it matures. So the one aspect of the maturation is the arrival of this modularity. So at the very beginning you can have like very less modular network, is more like together is a very coarse separation. One is like sensory and the other one is action, one is input, one is output, and then you start getting like separations between like this different sensory apparatus versus like head movement, body movement and motor outputs. And as the worm progresses, now you start seeing these different types of modularities that are coming arising. So where different subcircuits are getting rewired, so they are now like they're not like more uniformly connected. There are biased connections that are arising between different networks, different neurons, and that gives you this modularity. Okay, so what people realize that the neurons that form circuits in the mature nervous system do not share obvious relationship, meaning that there are some relationship but it's not concrete, because there are like I'll just try to highlight like two papers that came from Sitabra's group, and that talked about like this arisal of this subcircuits in the system using C. elegans, and they talked about like in a theoretical framework they talked about like different aspects of like birth order, lineage relationship, like distance between two cell bodies, how these subcircuits can bring together two neurons like that are far in the three-dimensional space but has like projections together, and also like that they're like sorry, I mentioned birth order, like so these many aspects they covered, and so there's some relationship. So what we didn't touch upon that. So what we did is what we realized that that many times the neurons that form these circuits, they tend to express certain transcription factors. So here like maybe more how many of you are familiar with transcription factors? So okay, so transcription factors are like crucial proteins that are that drives gene expression. So if you want to like so genes or the proteins, they are the driving factors for anything that happens in the cell. So if you have a certain transcription factors, meaning that they will probably express a similar set of genes, and that might give you that circuits. So what we did is we took like several of these transcription factors and look at how many of these neurons are expressing a single type of transcription factors. Let's take this example of UNC42, which is a very conserved, the name do not go by the name, this name can be ABC. So UNC means that it came out of the Sidney Brenner screen, where when they mutated this gene, the worm was not producing a coordinated movement. So what we found that this transcription factor is expressed in 40 different neurons, and so what we did is that we randomized the network and keeping the membrane adjacency, so the probability of getting connected intact. So if we take a random network that all has like interconnect like membrane adjacency, the ones that are expressing UNC42 tend to be connected, synaptically connected more than the randomized network. So there's a higher probability of connection, and these 8 transcription factors that all gave this high probability of getting interconnected or being in a circuit. There are also like I'm not going to mention here, but there are some transcription factors that always like tend to be expressed in neurons that are not going to be connected. So there is also a negative bias for some transcription factors. So but what is very interesting is that this set of 40 neurons that are connected by this in a circuit and all expresses UNC42 are not necessarily interconnected. They come from different lineages. They have like there can be interneuron, motor neuron, sensory neuron. they have like different neurotransmitter identities, but they're all, that is common is like they are all connected by synapses. So what we look at is like we took the connectome and made the subcircuit that all expresses UNC42, and you can see they're all very heavily connected all the way from sensory neurons to the motor neurons, and they're very heavy connections. And if you do a volumetric projections of all these neurons that expresses UNC42, they all project in the same neuropil, meaning they're all somehow, although they're very different, in their origin, they all come together and project in the same neuropil, which facilitates their interconnectedness. But again, if you take these random neurons that do not express UNC42 but project in the same neuropil, there is less tendency of making connections. So the adjacency or the bringing together is not the sole factor. Yes. Okay, sorry. Meaning that this neuropil means like this is a ganglia where this projection where all the nerve bundles are together. Okay. So if you don't understand, like just stop me. Now it's clear, right? Okay. Yeah.
I'm just curious. Is this a cause or effect? So, like for example, is it because they're connected that they're all kind of somehow expressing?
No, I'll show you the next slide. Yeah. So what MLA did, is a graduate student in a postdoc lab. So what MLA did, so there was some, so Sidney Brenner not only like in that time they not only did the wild type connectome, they also sectioned many of the mutant connectomes, but they didn't do the reconstitution because that one worm took 15 years, so they couldn't afford it. So these UNC42 mutant sections were there present. So what MLA did, she now like remapped this connectome in the UNC42 mutant. So this is old connectome data that is coming from the volumetric projection data that come from the Sidney Brenner's connectome, and this is a new connectome in UNC42. And you can see that if you mutate UNC42, this nerve bundle is not together. So this adjacency is lost. So and if you look at this two neurons, just two neurons, you see these two neurons are beautifully coming and projecting along over another. And now in the UNC42, this is a three-dimensional projection, but if you look at the 3D view, they're much more separated. So this is a structural aspect, but also if you look at the neurotransmitters that are the language how these neurons should talk to each other. The red means like cholinergic neurotransmitter, and the blue is the glutamatergic neurotransmitter. All of them in absence of UNC42 lose this neurotransmitter identity. And not only I'm not showing the data here, but not only the neurotransmitters, but also if you look at neuropeptides, gap junction genes, monoamines, like whatever way you can think that neurons can connect together, they're all getting very heavily dampened in UNC42 mutant. So I think that answers your question. It could be like a causative thing.
Well, I mean, I could be the devil's advocate and say that it's something else is causing their disconnection, and that is what is not causing them not to express.
So sometimes you see that if you look at like cell adhesion molecules, a mutant of cell adhesion molecules, the nerve bundles means where they're projected together, they gets defasciculated, means get separated, but they retain the neurotransmitter expression. So it's not necessarily if you lose the input they will lose neurotransmitter identity, which is little bit different from vertebrate brain, because in vertebrate brain if you lose the input, most of the neurons generally die because their survival is very much dependent on the activity. But in C. elegans, if you take this mutant and this conserved transcription factor, the Shubha Tole's lab also use some of these transcription factors. If you mutate those transcription factors in mouse, the neurons die, but in C. elegans they stay, although they lose all these characteristics. So that's the difference between invertebrate and vertebrate. So I think it is a causative thing, but and that also true for other transcription factors, but obviously you like we didn't do just silencing the activity in presence of UNC42 and see whether other features have done, but that data exists from other experiments where not UNC42, but if you lose the neuronal activity, they do not necessarily change the neighborhood or like neurotransmitter identity. So probably there is a causal relationship here.
So also I noticed that
Yeah.
the nature of the ASH neuron and AUB neuron seems to be getting changed, because you had a circle there and you had a triangle there. I thought the circles
So-
and the triangles represent the nature of the neuron, right?
Yeah, I'm not sure why is there. I have to, because the ASH is supposed to be a triangle because that's a sensory neuron.
Okay, but
So
you know,
probably here is it probably is misannotated here, because triangles are generally meant to be for sensory neurons. Yeah. So I think this is one of the factors that plays along with the birth order and the other theoretical work that came from Sitabru's lab and other people's lab, that gives rise to this generation of these modules. Okay, now this is just a single module that I talked about in ANC42, but in C. elegans you have, or in any nervous system you have different modules that are overlapping or talk to each other. So how that happens? So if you look at ANC42 itself, okay, so this is the entire ANC42 circuit. This is a but it also has subdivisions between it. So this is sort of like here is the sensory circuit, this is a locomotory circuit, these are all command interneurons, these are all motor neurons. So there you can also subdivide this circuit in different modules. So what is really fascinating is like what we found here. I don't have the real data to support that whether it's causal or circumstantial, but what it looks like, ANC42 partners with different transcription factors to even subspecify these modules, this circuit. So suppose like you have ANC42 working with this FAX1, it's another transcription factor to gives rise to these things. So if you lose ANC42, the entire identity of the circuit is lost, but if you lose FAX1, only this part of the circuit is lost, but the others are retained. So there is so then now in combination you can generate many more diversity using like a handful of transcription factor if you put like a combinatorial code. And this is what it looks like in different as a whole network, is a part of the network, where you can see that these blue ones are all motor neuron circuits. So they all express this transcription factor ANC3. There is another one that's green, they are also expressing this another transcription factors, and the red one is ANC42 that I mentioned. So there are different modules that are probably getting defined by different transcription factors come together to give you a much more larger scale module to give you the functional output. Okay. So now this is what we have in the hardwired connectome. But as I mentioned here yesterday, that the hardwired connectome only gives you a roadmap and gives you how the information can flow, but it doesn't guarantee that it will flow like that. So but also like what I was mentioned talking to Harish, like that the time delay information is missing. Like if you have a intersection where cars are coming from different directions, if they are all approaching at the same time, there would be collision, but it goes seamlessly because the cars are not approaching at the intersection all at the same time. There is a time delay. So you can, if you factor in that in the network, you can also get lot of these things. So that information is also missing from the hardwired connectome. So what people have tried, like what I'm going to show you today, like just example from one group, and there are other people also trying to do that. So what people have what is missing here is like you need to look at the activity of the connectome as a whole, and you need to identify which neuron is doing these activities. So at that time you can just say like okay, so I know that A is connected to B, B is connected to C. If I look at the activity, I should see that A gets stimulated first, then B, then C. But do I see the same pattern, or I see that A is getting stimulated and now C is getting stimulated, skipping B in the middle? So is there any deviation between the hardwired connectome and the actual real life activity? So to do that, you need to know A, B, C. You need to monitor all these neurons in a growing, behaving worm, and you need to monitor their activity at the same time. So for that, you need to have a very good microscope where you can monitor all the neurons at the same time, and you have to label them separately by different colors or different things, whatever way you want to label them. So I know that this is my A, this is my B, this is my C, and you need to also have the activity at the same time. So I can see that, okay, if I put a stimulus, what is happening at the same time in the entire circuit, and what is how the information is flowing, which way it is supposed to flow and which way is actually flowing. So to do that, like you need a powerful microscope, you need an activity sensor, and you need an identity marker, which I mentioned. So what this guy Ev Yemeni in my postdoc lab he did, it was a humongous challenge. So these are if you look at the head of the worm, so these are all the neuronal cell bodies like nuclei. And so how do you differentiate, how do you put label A, B, C in that? So what he did, he took 5 different colors, they all have like spectral separation. This is blue, this is GCamp, which is green but is a neuronal activity marker. How many of you know GCamp? Okay, GCamp is so every neuron that gets stimulated, it has more calcium in the cell body. Okay, so that's inevitable that you can use calcium, the amount of calcium in the cell body, as a proxy for neuronal activation. And GCamp is a dye that changes its colors based on how much calcium is there. So it is green, but if you put more calcium, it becomes brighter. So it's GCamp, it is there to monitor the neuronal activity. Then you have 3 different other colors that are spectrally separable. So you can, I'm not going to go into the properties of these colors, and there are many different dyes, like he used many of them to optimize which one is there, how many colors you actually need, how many colors you can actually reliably monitor in the microscope at the same time. And you can, so he could, and which neurons need to be colored in which color. So it's a humongous task, and so he put all these colors in different cell types. So what the end product is like at a defined, you can actually identify each of these single neurons, like each of these 302 neurons, based on their colors and their position. So like in this ganglia, means this neighborhood, only one neuron will have one certain color, and you can say that this is, if you look at the microscope, this is this neuron, I know that, and now let's see how it behaves in the function. So this is what it looks like in the worm. So you can see that there are different ganglia, means there are different neighborhoods. You can have like a same color in the different neighborhood, but not in the same neighborhood. But like you have this cyan color here that is not present in this neighborhood, but could be present in this neighborhood. So you can actually identify each of these neurons like in a very precise manner. So this is how it looks in a 3D projection, that this is the head of the worm, and you know exactly which neurons are present where, and you can define these neurons. Now let's look at what.
Which is the head?
So this is the head. This is the head. The blank space is the pharynx. So this is the nerve ring. So sorry. So this is the nerve ring. This blank space. So
Why are you not seeing the nerve?
Because these are all nuclear reporters. So you have like 2 different ganglia, like this and this. So if you draw the worm like this, and the pharynx is like this, very bad drawing, but you have certain new ganglia. This is the anterior ganglia is there, like this anterior part. Then you have this posterior ganglia, they are more heavy, and these all neurons are projecting in the nerve ring. So this area is devoid of any cell body. So this all like neurites, like the axons.
Only showing the cell cell body position.
Yeah, because the problem is if you do that for the cytoplasm, if you see a green color here, you don't know whether it's coming from this neuron or this neuron. You cannot disambiguate that. So that's why it's only it's limited because you don't have the cell body information. But this is what you can do at best.
So what's the green blob at the tail?
Oh, sorry. So this is an artifact. This is a another cell that is supposed that somehow is getting expressing this one of the colors, but.
And there are lots of tiny.
So these tiny dots are actually like they are autofluorescent granules. So in C. elegans you see a lot of autofluorescent granules that are like these are protein aggregates. They generally tend to fluoresce under UV or like in the higher wave more energy lasers. So that's why you see it in the green but not with the red. So yeah, but we know exactly like this is not going to be a nucleus because the size is very different and these are too tiny to be a nucleus. So we can do that.
And are these all the 302?
Yes, okay. Except two. So there's 300. There is one neuron called CAN. It's a very interesting neuron. It's a neuron. It expresses all the neuronal characteristics when the worm is born. After L1, it stops expressing neuronal markers. It becomes like a dormant type of cell, but its projection is there, and in the very earlier on, it makes neuronal connections. So at this level, none of the neuronal reporters are expressed in the CAN, and they do not make connections at that stage. So it's 300. So this is what happens. Yeah. Yeah.
How's the color assigned to each neuron?
Oh, it's genetically encoded. So each color is, so suppose GFP is driven under certain neuron specific promoters, RFP is driven under certain neuron specific promoters. So you see a lot of these like these are like true colors and some of them are in combination.
So you get a unique combination.
You get a unique combination. Yeah.
So if you had to design that.
Yeah, he designed it. So he designed all of it, and
so it was designed with
keeping in mind
that two neurons should not express the same color in the same level.
Exactly. Yeah. Yeah. So this is what happens if you look at the whole head activity. So you can see these neurons are now fluctuating, means that these neurons is showing variable activity. Now there is one. So look at like there's other chemical names will come up here. So this is the basal state of the neuronal activity, and this chemical when you come, these neurons are like now fluorescing much brighter, means that these neurons are showing more activity under this specific stimulus. And you can put NaCl and there's a different set of neurons get fluoresce. If you put another thing, it gives you different fluorescence. So now you can look at like in all these minute variabilities at the ground state, and you can see that at a ground state when the worm is not behaving just like the inherent activity of the nervous system, is there any rhythmicity between different neuronal activities, and how under certain under certain stimulus how these neurons are behaving. Okay, so now if I just give you an example, like just suppose like this neuron is, we know that is connected to this neuron. So we can ask these questions with this data, that when this neuron gets stimulated, does it always channel its activity to the other neuron or something else is happening? If it's happening, fine, that means the hardwired connectome is predicting the path. If it's not, then what is wrong? Why is not going into the defined pathway? Can we do some experiments to figure it out? Okay, so now in collaboration with other labs, like Eve also designed a machine learning algorithm where you can actually take a picture of these worms, and this algorithm like straightens it up and automatically gives the cell ID and you can actually predict how the neurons behave, like so this is semi automated. There's also like some flaws are there, but you can actually give you there's a good automated pipeline how you can image, take the picture and do the calculations. Okay, so the what data came from and so okay, so now you can look at this neuronal traces. So you can actually see in the right panel that we can measure all these different single neurons, how their activity is happening under different stimulus conditions. And you can also project it in this way, and you can see that under NSCL this set of neurons are getting upregulated, where in the other stimulus this set of neurons are getting upregulated. But the funny thing is, like all of them are attractive odors. So they are all generating warm attraction, but through a completely different set of neuronal subsets, the subcircuits. Okay, and also if you don't challenge it with any particular odor, if you just look at the inherent variabilities, you can now see that these neurons are showing like periodic oscillations. So we didn't have any idea that warm neurons can show this kind of periodic oscillations, which people have figured out in mammals, and they show these different oscillatory behaviors. So you can actually measure the oscillation frequency, and you can also do it with a freely moving worm, and you can measure that these oscillations are somehow matching with the crawling frequency. So probably this has something to do with the crawling of the worm. So what I mentioned, what they figured out is that if you take the circuits, like how these different attractive odors project, like if you take like which neurons are getting stimulated by different odors, and if you project them in a 3-dimensional space, you can see that although all of them are showing the same attractive behavior, they project in a different 3-dimensional space. So means they're separable. So but ultimately if you look at the output, that it all produces forward biased locomotion towards the source of the odor. So now you can actually do it like with real time data, that this is my hardwired connectome, this is my like real connectome, does it match?
So I'm just wondering, like does that mean that you can actually work out the olfactory code for C. elegans? Because like this is what Linda Buck did for mammals, but for C. elegans I'm always amazed like how using so few neurons they can actually do an olfactory code.
I think it's difficult, because if you look at the vertebrate all the way up from insects, like the olfactory neurons are they're separated in space.
Yeah.
So they all express like receptor A, B and C, and they all like project to different glomeruli. But in C. elegans the same neurons can express many different olfactory receptors, but the surprising thing is like they do not generate the same behavior. How? I have no idea. So it's supposed to be, I mean if you are stimulating the same olfactory receptor, it should generate the same behavior, but it's not. And actually you can do like so it's it gets more obvious when you do a choice assay. Like suppose you have you can put like NSCL and another odor in 2 different spaces, and can you ask the worm whether you should go this way or that way. If it's like if it's stimulating the same olfactory receptor, it should not be able to discriminate, right? But it can discriminate. And the other interesting thing is you can train the worm by doing some associative learning, by like Pavlovian conditioning. So you can pair an attractive odor with starvation, and now the worm learns to avoid that odor, just like Pavlovian conditioning. So same neurons, same odor that stimulates the same neuron, you can differentially condition them. So there must be a way to separate it. I have no idea, and it's still puzzling how the worm does it. So it, all I can think of is like, that the downstream signaling pathways are different, although they are both GPCRs, they both activate the similar sort of downstream signaling pathways, but could be the there is also like, in the minute details, like, in the GPCRs, like, what type of G proteins you are using to channel the signal, that might be different. So the signal, although both of them are similar type of receptors, the second messenger that are coming may not be similar. So that is how the distinction could be made, but it is a very fine distinction, we still do not understand how they do it. Okay. So so I showed that there is a pathway. Now you can see that this is my functional pathway, and you can see that this is my hardwired connectome. Does it match? It does not. So if you look at the correlation between the head activity and the connectome, is very loosely correlated. So the chemical synapse is even less, and the electrical synapse is somewhat little better, but is still not the 100% correlation. The tail activities are all very similar. So where are we losing the information? So if it's only can be predicted by the hardwired connectome, it should match. So here I think I want to talk about this part of the connectome. So the hardwired connectome, you can see these two things, but not this. This is like a monoaminergic signaling. So you have all heard about like dopamine, serotonin, there is also tyramine, octopamine. These are all like chemicals that are secreted in our brain, very conserved from nematodes, and they all generate like our mood, like modulate our mood, activity, and lot of these things. They do not like form this kind of synapses. They can work like over distance, and also neuropeptides. So neuropeptides also can go from one place to and get secreted and just does another things, like neuropeptides also like insulin you all heard about, that's also like work over long distances. The other thing you probably have heard in the high school biology books, like this oxytocin, vasopressin, and those are also neuropeptides that are very conserved, that do a lot of this kind of modulations. Okay. So people have tried to do it, is very difficult to do it, because what you have to do is, now you cannot do an EM and look at like this is going to be my connectome. So what you have to do, you have to look at what monoamine can bind to what receptor, and you have to identify the neurons that are expressing a certain type of monoamines like dopamine, and you have to look at like what are the neurons that are expressing dopamine receptors, and you have to make a circuit out of that. So you have to do it's a lot of work to identify like which neurons that are expressing, which neurons that are receiving, and it also does not tell you that although they have this receptor ligand pair, they are actually connected. Okay. So this is how it looks like. This is a work from Bill Schaefer's group, and you can see that these two neurons, this RIM-R and RIM-L, these two neurons, only these two neurons in C. elegans expresses tyramine, and these arrows suggest their hardwired connection. So they are connected to all these different neurons, but if you look at like the neurons that are expressing tyramine receptors are all these ones that are shaded in gray. So there is no correlation between hardwired connectome and the monoamine connectome. Again, there is another example. These two neurons RIC and L and R, the left and right, they express octopamine, only two neurons, and these are the neurons that it connects to in a hardwired fashion, and none of these hardwired connected neurons expresses octopamine receptors. All the ones that expresses are separated. Okay. So they mostly work in non-synaptic, non-hardwired synaptic fashion. Okay. So, what you can get if you do the topology, you can get like they're very very centralized, sort of like they give you that hub type of structure, and where a few of these neurons that expresses monoamines talk to lot of these neurons. So, that are present in all over the body. So, you can, so this is the statistics, like that only 18 of these 302 neurons expresses monoamines, while 251 of them expresses the receptors. So, you can see the extent of this connectome, but and it's completely different from hardwired connectome. So, if you look at that, all of these three modes of connections, like monoamines, there are four actually. I am leaving out neuropeptides to talk about it in the next part. So, if you take like the intermotor and sensory neurons, and this is the pharyngeal neurons, and if you see how they are connected with gap junctions or electrical synapses or chemical synapses or monoamine synapses, and there is no correlation actually. So, one thing that stands out, that monoamines give you a direct access from sensory neuron to the motor layer. So, if you look at the gap junctions and the chemical synapses, there are very few connections between sensory and motor layers, direct connection, but monoamines give you that that advantage. So, it reduces the distance and it breaks down that modularity. So, sometimes it is good because it reduces the, I think I have the, okay. So, this is how it looks like if you project it in a different way. So, here this is the interneuron axis, this is a motor neuron, this is the sensory neuron axis, and the hub neurons, means that are more connected, are away from the central point. And if you see that these monoamines are actually mostly radiating from this hub, so called hub neurons, and this connection, this axis, this connection between sensory and motor neurons are very much predominated by this monoamine signaling, but not with the hardwired connectome. Okay.
So, how do I intuitively understand the monoaminergic signaling network? Like, for example, synaptic network, I can understand that it could be, for example, rectifying and like it can give both activatory and inhibitory kind of connections. Gap junctions tend to be only excitatory and they're faster. How do I intuitively think of the monoamine network as contributing to the communication?
So, I think, I mean, I don't have like complete answer, but what I think that in certain cases, like in case of say if you want to avoid a predator or some things like that, you want to jump like from sensory to the motor layer. So, that kind of situations probably like reducing the degree of connectivity could give you advantage. Okay. So, or you want to bring two different circuits together. Okay. So, like I will give you one example. So, the worm actually like makes this sinusoidal movements, they go through that. So, there is a natural predator of worms, is there are some fungi that makes this loop like hyphae. So, they are kind of literally kind of like makes this loop like hyphae. So, once the worm moves through that, this wiggling motion triggers this noose to close. So, they get captured and then they get digested and the fungus eats it. So, as soon as this worm touches any obstacle that are very low threshold touch, this tyramine signaling kicks in, that RIM neuron gets kicks in. So, now you have two different circuits. So, this in a worm you have separate circuit for head movement and you have separate circuit for body wall movement, for making postures. So, in this case tyramine, what it does, it brings all of them together. So, now you at the same time what it does, it stiffens his head. So, it is not making movement. So, it is not supposed to trigger the noose and is making a backward locomotion. So, both of these happens together. So, otherwise they have like separate circuit, but this tyramine converging both the circuits to make a simple behavior. So I think this kind of situations could be that's useful when the monoamines are there, but otherwise you don't want that to happen, because you want like separate circuit for head movement and the motion, but in certain situations you need to break down that asymmetry and degree of connectivity. Yeah, so I think that could be a reason.
So would you say that it's something like the reflex, it is behaving something like the reflex of the elegans, in the sense that it's short circuiting the sensory to the motor direction?
I think so. I mean, it looks like that. Like, you know, it's a short circuit between sensory to motor neurons. So yes, I mean, in a way that it's, but it's also like, you know, modulatory, because you don't have it all the time, I guess. There is no way we can measure it, because the receptor and the neuropeptide is the monoamines are expressed, but whether they're released at the same time, that we don't know. But that also, the release is also triggered by the hardwired connectome. So there is a way that certain circumstances you need to short circuit the brain and to generate a one behavior. So I think that could be the reason. Yes.
So you know, like for example, how does the monoamine actually work on the elegans neuron? So let's say you have a sensory neuron which is producing this monoamine.
Yes.
How would it work on, let's say, one motor neuron? Is it like diffusing or?
Yeah. So it diffuses, but we don't know actually like, you know, how far it can diffuse. So so that's a gap, because like, you know, if you look at the but generally I'm not sure about the monoamines, but generally the neuropeptides, which is kind of similar, they're more, the hub neurons tend to have very long neurites.
Yeah.
So they can generate they can secrete it over a very long distance. So monoamines could be something like that, but I think their diffusion is not that much, because I think they're more like, you know, local than neuropeptides. Yeah.
So you're saying that it could be that the neurite of the sensory neuron, which is closest to the cell body of the.
They're not necessarily sensory neurons. There are also interneurons that.
Right. Suppose you have a sensory neuron. It is basically emitting a monoamine close to the cell body of the motor neuron that they wish to activate, and that monoamine is going and binding to the motor
receptor.
neuron.
Yes. Yeah. Yes.
What does what do they do to the at an individual neuron level? Say receiving one of these monoamines, what does it exactly do to the function of the neuron? Does it change the, say, like for example, the resistance values, or like,
Yes.
what does it do?
Yes.
It changes the constants.
Yeah, it changes so it has a receptor called GPCR. So when it binds, that opens up the ion channels, that changes the membrane potential and activates or inhibits the neuron, depending on which receptor it binds to. If it binds like, you know, chloride channel, it will make it more negative and inhibit the neuron. If it's like, you know, binding to a cation channel, then it will like, you know, activate the neuron. So if you look at like, you know, that that what happens with the monoamines, so this is the path length you can measure, like, you know, between two different neurons. So this is the synapse, the chemical synapse, and this is the gap junction or electrical synapse path length. If you take any random pair and measure like, you know, what is the degree of separation between these two neurons, and the monoamines have very less path length. So they offer a direct mode of communication. Also, like, you know, what we talked about, like, you know, the modularity or the subcircuit specification, the chemical synapses on the gap junctions, they offer like a high amount of modularity or subcircuit, but monoamines, they offer very less modularity, because so they're kind of like, you know, trying to unify the whole connectome. Okay. So there's also another type of non-hardwired connectome, which is called neuropeptide connectome. It's like it's a humongous challenge, because again what you have to do, that you have to identify which neurons expresses neuropeptides, which neurons expresses neuropeptide receptors, but at the first level you need to identify which neuropeptide can bind to which receptor. So, if you take all these neuropeptides and receptor combination, it comes down to around like 55,000 potential combination. Okay. So, so there is a paper that the group in Belgium, they do it, and they have a robotics and pharmacological method to test all these potential neuropeptide receptor combinations. So, they come down to only this 600 and 450 odd, like 60 odd neuropeptide receptor pairs can actually function in a meaningful way, meaning that they show enough activity at a physiological concentration of neuropeptide. If you give lot of neuropeptides, all the receptors might give you something, but like a physiological concentration, these could be the meaningful neuropeptides, and they publish this, and they have this we know that exactly which neuron is expressing these neuropeptides, around like 70, 80 odd neuropeptides, and the receptors we know exactly where they are expressed, and we can do a map. So, this is same group like Isabel Beat's lab. Now we also have RNA-seq data to validate where these neuropeptides and receptors can be expressed, and this is how it looks like. So, it's basically all the neurons in the C. elegans can be connected by neuropeptide signals. Okay. So, this is all neurons from this giant ball of neuropeptide connectome. So, here you have like the radius of this circle. The bigger the radius means that this neuron is connected to another neuron by many neuropeptide receptor pairs. So, you can say that so okay, if you take this neuron, the small circle means that this is connected to its partners only say by 5 different neuropeptide receptor pairs, and this neuron that is very like really big circle means that it can connect to its different neighbors by probably say 50 different neuropeptide receptor pairs. So, it has like that is how it's more weight is given to this neuron, and these are kind of acting as a hub neurons in the neuropeptides circuit. If you look at like neuropeptide like topology of the neuropeptide connectome, it can come in many different forms. So, you can also have local. The open circles here means the ones that are secreting neuropeptides, and the solid circle means that can potentially receive. I mean, we do not know whether they are receiving, but they have the potential receptor to receive to this neuropeptide. You can also have a pervasive type of network where there are many neurons that are secreting neuropeptides and many are receiving. Also, you have a broadcasting type of neuropeptide architecture. So, very few of them are expressing neuropeptides and a lot of them are receiving, or you can also have like an integrative neuropeptide signaling. So, there are lot of these cells are expressing a certain type of neuropeptides, but only a few cells can receive it. There is also other interesting thing is that there is no one-to-one correlation here. So, there are also versatile neuropeptides that can bind to more than one receptor, but also there are like promiscuous receptors that can bind to different neuropeptides. So, it is literally a mess, and I will give you one example that we all somehow familiar with the insulin signaling, right. So, there is a single insulin receptor in C. elegans, and there are 30 odd insulin like neuropeptides. Some neuropeptides, insulin like same family of neuropeptides, when they bind to the same receptor can activate the neuron, the others can inhibit the neuron. Okay. And there is, if you look at the structure, if you look at the evolutionary phylogenetic tree, there is no way you can predict whether it is going to be inhibiting or activating. So, it is very difficult to work with these neuropeptide receptors, and it is always scary. Okay. So, here the same, these different graphs are showing the similar type of architecture, but again there is one problem. if you look at the worm, all these neurons that when we look at this this kind of ball, the positional information is lost, but they are not all are congregated together. They are 3-dimensional, they are separated in 3-dimensional space. The cell bodies are separated in 3-dimensional space, and the neurites that are projections are also separated in 3-dimensional space. So, 2 neurons can have cell body in very different places, like in vertebrate brain or in C. elegans brain or wherever you take it, but the ultimate thing is whether they are projecting to the same place, whether they make connections in the brain. So, if you look at this C. elegans architecture, these neurons are separated in different ganglia, and now you can say that okay, my neuropeptides, do they act short range, means within the same ganglia, or like in the medium range between the neighboring ganglia, or they can also act in a very long range. Okay. Different neuropeptides, they have different diffusion coefficient, they will diffuse very differently, they will also get degraded, their degradation kinetics is also very different. So, you have to factor in all of it to understand whether a neuropeptide might be that is released here might be acting here, or they should be like acting locally. But in a more conservative fashion is we can say that okay, the local connections must be happening because they are so short, the neuropeptide should reach there, and somewhat the medium range connections should be happening. The long range connections we do not know, but there are enough experimental evidences that suggest that in some cases the long range connections can also happen, but you have to test one by one empirically. And so, this is what it looks like in a all if I like summarize all of it. Okay. So, these are the different neurons, all the different neurons that C. elegans has, and you can see that this is like motor neurons, interneurons, sensory neurons, and based on like what kind of neuropeptide receptors they express. And here there are the same neurons based on like sorry, here what are the neuropeptides they are expressing and what are the neuropeptide receptors they are expressing. And if we put all the data that has been validated interactions in this diagram, it comes to a complete there is no separation. So, it is a very singular dense network, you do not get modularity out of this thing. And if you look at like in any single place like this where the authors highlighted these two neuron pairs, you can see like so many different neuropeptides this neuron is expressing, and there are different neuropeptide receptors that are also this neuron is expressing. So, they can talk in a very different way, or at the simultaneously they can use like multiple different language to connect to. And these are all like blue, or these colored ones are all like short range signaling. If you only take the short range signaling, you can get that much variability. If you also incorporate some of the medium range interactions, you can also get some other interactions, meaning the combinations are extremely diverse. So, all I want to like want you guys to remember that there are not only the hardwired connectome, but there are also like non-hardwired connectome that do not follow the simple like modularity principles that we see in the hardwired connectome. They can be very very integrative, they can also have promiscuity, but so you have to be very careful when you interpret the hardwired connectome data and the functional connectome data. So, what you get from the human brain, what you get from the MRI is a functional connectome. That does not mean that they are physically connected, because the two lobes can be very different, very distant. They do show like connectivity in the functional MRI, but they can be communicating through neuropeptides or monoamines. So, if you have to take both of these things together and then make your rational conclusion out of it. Okay, so I will stop here, and I think you guys will need a coffee, and we can again congregate to have like a gap junction. Okay.